I'm quite convinced, and I think we've been pushing ourselves to be convinced that manifolds are the ideal level to look at neural function. But I don't know if we will have an understandable mapping from neural function to behavior. There was a lot of argument, more about, is this concept useful? And I think now we have enough evidence for that. Maybe a bit too much, because I feel like science should be more like confrontational, in the sense that we have more of these discussions. Now there's not those types of counter arguments. It's more like, how is this not trivial? Like, of course you find this, what have we learned? By comparing recordings from monkeys doing reaching and grasping tasks and mice doing reaching and grasping tasks. And we basically showed that these manifolds were similar across different monkeys or different mice, and the degree of similarity depended on how similar their movements were.
This is Brain Inspired, powered by The Transmitter.
Hello, people. Welcome to Brain Inspired. That is Juan Gallego. Juan runs the B-Neural Lab, as in Behavioral and Neural Dynamics, at the Champalimaud Center for the Unknown in Lisbon, Portugal, which is probably the most awesome name for a center that I've come across. They probably know that. Sorry to splice this in here, but just a quick correction. Juan has renamed his lab to the Neurocybernetics Lab and wanted me to note that they are affiliated with the Neuroscience of Disease and Neuroscience programs and the Center of Restorative Neurotechnology. Okay, back to the original recording. The main reason I invited Juan is because he has worked a lot on neural manifolds, the mathematical objects that neuroscience is using more and more to describe how big populations of neurons coordinate their activity to do useful things. In fact, he recently gave a short talk that he titled The Manifold Manifesto, because he was asked to be provocative. And he was provocative, suggesting that manifolds are real as chairs and tables are, that they have causal power, and that they might be a target of evolution. Very provocative. Of course, he talked about his own work and others' work to support those claims. So today we discuss many of those themes through the lens of his own work and others' work. And we talk about what keeps him up at night, about the possible limits of using manifolds to connect brain activity with behavior and with mental phenomena. Juan is not just a manifold person, though. He's more broadly interested in motor control and how brains do it. In that vein, we also discuss his work in patients with spinal cord injuries who don't have enough nerve connections to their muscles from their brains. They don't have enough connections to actually enact their muscles, to actually move, but they do have enough nerve connections so that some signal still gets through. And Juan and his colleagues can detect that little bit getting through and use it to infer what behaviors the patients intend to do. And they can use that information to control actions in a computer simulation. The hope is that this will translate to controlling prosthetics to give spinal cord injury patients their mobility again. If you're a Patreon supporter, this is a super long episode because I coaxed Juan back on to have another short conversation to discuss a few other topics that we didn't get to. And I'm including a little snippet of that extra discussion here, but go to braininspired.co to become a Patreon supporter for the rest of it and to get the full versions of all the episodes. So thank you to my Patreon supporters as always, and thank you to the Transmitter for also helping support Brain Inspired. Go to the show notes at braininspired.co/podcast/234, where I link to many of the papers that we discuss. All right, I hope you and your manifolds are doing well out there. Here's Juan. Juan, we're going to talk a lot about neural manifolds today, among other things. And when I think of you now, I synonymously think of like manifolds. Is that an accurate thing? Or do you even like that? Do you think that's true? And do you like that?
I think it's maybe I'm part-time manifold, but I think I'm also other things I want to believe. But I guess we've been try to be opinionated about what a manifold is and also try to clarify the pros and challenges of this framework. So I guess it's fair to some extent.
Yeah. Well, yeah, okay. Pros and challenges. Let's just jump right in, and I'm going to ask you to do something that you've probably done a lot, but what is a neural manifold to you, perhaps, because I know that there are different definitions as you've written about. And then, so just broadly, and then how does the concept of a neural manifold fit within your thinking these days about brain function?
Yeah. So I think to me, a neural manifold is just like this mathematical object that captures the possible neural activity states given the constraints that a neural population has. And these constraints can be like, connectivity, neuromodulation. So all this, we call this in a recent paper, biophysical constraints, for lack of a better word. But of course, there's also the task constraints of what the animal is doing, right? So that is what I would call a neural manifold.
Well, so you already, oh man, now I just immediately want to go. So, but a neural manifold, let's say, is defined by the spiking activity of neurons, right?
Mm-hmm. Yeah.
In a population of neurons, which separates it from sort of the single neuron doctrine where the field used to really put a lot of focus on like the tuning properties of single neurons. And a manifold is a way to take like populations of neural activity, look at how they co-vary with each other, and then come up with some sort of structure to describe how the population of activity relates to ongoing behavior and cognition, right?
Exactly. So I think what you said, I think covariance or coordination, to me, that is the key term, because a lot of people, I think there's a lot of emphasis on talking about manifolds as being low dimensional, right?
Mm-hmm.
But I think this is very unfair to everyone looking at tuning curves or the first single neuron recording, papers with single neuron recordings where basically people were building categories of neurons, right? Like these are ramping neurons, tonic neurons, or these are muscle cells, force cells, velocity cells in motor control, or play cells, edge cells, and all these things. So we were I think in neuroscience, we were always low dimensional. We're trying to build low dimensional classes because as philosophers say, we need like low dimensional objects or compressed concepts to reason with, right? But I think the important thing in manifolds is that you switch from what single neurons are doing to what the collective is doing as a whole. And then there's a lot of these things that the collective may be doing that cannot be mapped, at least very clearly, onto the constituent neurons. So that is, I think, what is the key distinction.
Well, part of that is that, so continuing on the theme of where neuroscience was in recording single neurons, I mean, the way that you would do it back in the day is lower an electrode and you'd sort of listen for a neuron, and that neuron, you wouldn't record it if it seemed kind of random or wasn't like modulated relative to whatever task your organism was performing. And so you would actually bypass a lot of neurons that you might think is noise, right? And sometimes the neuron that you end up recording for an hour or 20 minutes or whatever it is, sometimes it's not modulated the way that you would expect it to be modulated, and it seems a little noisier. So then you end up with this kind of population of recorded neurons, and you have to think, all right, which one is relevant to the task that the animal was performing? Meanwhile, all the neurons are firing. Some of them you consider noise, some of them you consider somehow adjacently related to the task that you're performing. You have to like make these decisions. And these days, of course, we put an electrode down. We don't try to isolate any single the population approach. We don't try to isolate any single neurons. We just take them all. And this is a way to sort of collectively say, well, to include all those neurons that maybe we couldn't make heads or tails of in the past.
Exactly. No, I fully agree. And also I think thinking should evolve with technology, right? And technology unlocks like thinking and understanding, right? So, there were good reasons why people were doing it back in the day. Again, our textbooks were written like with these approaches, but I think that now that we can collect different kinds of neural data, we can think of different ways of making sense of neural data. And to me, the most important change, I think, from like enabled by this population approach was like, again, the first studies suggesting that the collective is more than the sum of the neurons. So the first paper, if you want me to give you a quick example, the paper that kind of made me go down this rabbit hole is a paper by a common friend, Aaron Batista, and Byron Yu, when in 2014 they published this paper basically showing that these collective patterns of activity constrain what a population can do. So I'll describe it very quickly for everyone in kind of lay terms. So this is a bit of a technical paper. So what they did was had monkeys doing a standard brain-computer interface task where they would map neural activity from populations onto the movement of a cursor on the horizontal and vertical axis, right? This is the typical VCI task. And what they did was like do this through a manifold. And their hypothesis was very nice, like very elegant. It's like if manifolds reflect constraints, learning something new within the manifolds should be easy, and learning something new outside the manifolds should be hard. And that is exactly what they found. So
let me just pause there and just clarify what inside and outside the manifold, or on or off the manifold is, right? So you have like this population of activity, and so the neurons collectively, individually, the neurons, they all have like ranges of their firing rates under different conditions, for example. And if you think of like, if you have 10 neurons, you can think of the state space of the neuron, of those 10 neurons as like a 10-dimensional space where the neuron can go from 0 to 100 spikes per second or whatever. So that defines the total space of possible trajectories of that neural activity, right? But the manifold is defined, and you mentioned low dimensional earlier, is in reality the patterns of the population of the individual neurons collectively that make up the population only visit like certain parts of those state spaces. And often what we have found, especially in motor related activity in places like the motor cortex, is that the range of possible spaces within that state space that the population activity actually visits is fairly limited or constrained. So in a normal task, let's say like an animal performing this cursor mapping task, you'll see that like there is kind of a low dimensional structure to the population of activity while the monkey or animal performs the task. And I think what you're saying is like when they change the task, but keep it within the range of that well-defined manifold, then it's easier to, I forget you should jump in now and correct me.
Yeah, no, this was a much less compressed explanation of this. So what I was saying is after doing like this, finding this low dimensional manifold that Paul was describing, that is what they observed these neurons were doing when the monkey was doing the task. It was easier for monkeys to generate new patterns of activity within this surface. So respecting, if you want, like within the patterns of working together that they had observed, then change the firing rates a similar degree, but asking the neurons to work together in a different way. So it's about, in a way, neurons could do different things easily if they were working together the same way. But if you ask the neurons to do a different thing, working in a different way, they couldn't. I hope that was clear.
Yeah. No, that's great. And that's what got you into the world of manifolds?
I think this paper and a couple other papers made me think that it was more than basically a nice way to look at data. Back in 2013, 2014, there were very fewer papers, right? Like this has, I'm sure if someone goes and Google Scholar, like this is skyrocketing, right? Yeah, but it was this idea of, these ideas of emergence that people have discussed a lot on the podcast that, that if you want like this coarse graining allows you to see things that maybe are not so easy to understand at a lower level.
Well, I saw you give a talk at a recent conference, and I know that you were kind of asked to be a little speculative, a little what's the word? to generate discussion. And I don't know if the title of the talk was the Manifold Manifesto, but that's the phrase that you use, which is great marketing, by the way, you know. But so in that talk, you were discussing. Well, so stepping back, like I kind of think I go back and forth about the importance and usefulness of the concept of manifold. In one sense, it's obviously beautiful. You get these ring attractor manifolds and these things that are geometrically satisfying to describe. And it's like a miracle that the population of these noisy neurons can covary in a way that maps onto these structures. But then also in my own recordings, I am, which is like in motor cortex, for example, in a mouse, I think, am I looking at a manifold or some like Swiss cheese or just like low level? Like, how should I really think of this as a manifold? Because it doesn't map out to those nice trajectories. So anyway, I mean, that's why I kind of at the beginning asked you to, where manifolds are in your thinking these days. Because you gave that talk and said Manifold Manifesto, and then listed some things that might be useful for manifolds. But I'm wondering like how convinced you are that they are ontologically real objects and how much we should be studying the manifold versus other things,
Yeah, no, that was a fun talk to prepare. And it was also like, I prepared it like the week after I moved here, I think. So we were unboxing and stuff, but it was fun to do. So I think, because I had also to compress, I focused on what I think manifolds are and what they mean. So the 3 points that I wanted to make in that talk, and we can go through them later, were first that manifolds are real, as in they are, invariant patterns that reflect real biological constraints. So they are ontologically real as chairs and tables are. That was the first point I wanted to make.
Yeah. Okay. No, I'll let you get through the 3 points because I want to jump in.
Of course. The second one was that they are good for compressed understanding, like we were talking. But in that sense, like tuning curves also were, right? But manifolds, as you were saying, like you see these beautiful ring attractors, and so you get these compressed descriptions that we can discuss whether they are useful or trivial. And which way to go, right? Depending on what you're looking at, I think is the answer. And the third point that I was trying to make is that I think they have causal power on the organism. So they are not only like, real, but like the manifold influences what the animal is doing. What I didn't have a lot of time to talk about is what I think are the two things that keep me awake at night with respect to manifolds. I only outlined them very quickly, which were whether we would be able to map the concepts like from, psychology or psychophysics that have taught us a lot into, this is, I'm playing Tetris. Oh, this means that I'm going to do this and this, so I'll have this attractor move to this point, right? And how to go between these two levels, the description, and the other level that I think applies to any way of looking at neural data is basically if we are not trapped into this mereological fallacy of saying like, basically that things that we do, like making decisions or initiating a movement or thinking about what we're going to cook this evening, can be mapped into a small part of the brain that we can understand. Even if I don't think there's, even if I think there's modularity in the brain. So I think these are the 3 good points and the 2 weaknesses.
Oh, okay. Yeah, okay. I mean, you've expanded on that last point more in a review, talking about so we have this phrenology background where we think area X does function Y, right? But one of the things that you've written about really nicely is that with the advent of like these modern technologies and recording in more naturalistic settings, and more complicated kinds of tasks, that there's more of a hierarchy view that like a brain area, and people's work like Eve Marder, et cetera, whom you cite frequently, a brain area can do different things depending on the context and depending on the complications of the task. And then, so the idea that like brain area X has a ring attractor, brain manifold, brain area Y, it has a slightly more nonlinear manifold. And then these two, and you just called them causal. So these two manifolds must interact with each other and be pushing and pulling from each other, and then somehow construct up to these psychological terms that we use. And so that's where your worry is like trying to like combine these things, or?
Exactly. Like, can we even do it, right? Like one thing that we were discussing in the last review, I think is the paper with my friends Matt Perick and David Kanarayan that came out, I think last year, but anyway.
This is the manifold view of the brain. Is that the title?
I think so. Yeah, I think that may be the title. We had a different title and got like changed in the last minute.
Okay.
So in that paper, I think we discussed it a lot among the three of us because we wrote this mostly on Zoom, which was what made it fun. Like what, because we all agree, I think I want to speak on their behalf, but I think we all agree that the brain is modular, right? And, like things like a stroke, like so that if you have a lesion in this area, the deficit will very special. You have a stroke here, you will have this type of issue, right? But the problem is that maybe this doesn't necessarily mean that this area is doing all that, right? It just may be part of this aggregate collective brain state that does that process. It just has like a focal effect on that process. This is what I meant about the neurological fallacy. So maybe there's no way that we humans can understand of mapping these human processes into how they're implemented in the brain, which is basically a discussion I often have with John Krakauer now in person these days. And that connects a bit with to their paper on mental representations with neural representations that I think I'd recommend people to read.
Yeah, well, I mean, on the other hand, I mean, it's like our best bet right now, right? Because it's an in-between. I mean, I think that's what John would argue as well, and has argued, is that these sorts of emergent structures, for lack of a better term, they're not single neurons. They do have shape and characteristics, and they describe well they're like a go-between, because you used the word constraint earlier in terms of how they're real entities and reflect the real constraints of the system, of the underlying like physiology, but also they reflect the constraints of the tasks that we're performing, that the behaviors and the psychological psychological outputs. So I mean, perhaps are the best thing that we have going right now anyway.
Yeah, I mean, that is what I think, right? And I think a lot of our work, first when I was a postdoc and then in the lab, has been like basically convincing ourselves that manifolds are real, in the sense of, finding invariances like over time during a learned behavior. So, no matter which neurons you are recording from, if you look at a brain of a monkey that knows how to do this task today, the manifold will look the same 2 years later, even if we know we are not recording from the same neurons. And the monkey has had an interesting life and learned new things and stuff. So, this was one of the examples or the studies that made us think that they are real, right? And we can talk about other examples, like, for example, you talked about the ring attractor manifold, right? That has been found in the thalamus, in the navigation system, right? So we know that this manifold is not only the task, it's not the mouse or the rat looking around, because when they go to sleep, the activity remains confined to this ring attractor, right?
Mm-hmm.
So this goes back to your point about constraints and why I think manifolds are real. And I do think they are the best thing we have, but I'm also trying to be critical with my own ideas. That said, as you suggested, I think it's not only this. I think they also have causal power, which we can talk about why I think that way. So it makes me feel even a bit more convinced that they are interesting.
But with the level of feeling convinced, if you're like me, the level of worry that I'm wrong also rises, right?
Exactly, right. But that is, I think, what science should be, right? Like you want to, if you want to push your beliefs and you want to disprove, at least I would like to be the one who proves me wrong if I'm wrong.
Yeah. Well, ideally, you don't have to talk about beliefs eventually.
Exactly.
Well, you mentioned the work that you did in talking about the invariances, and some of what you have done I mean, you've done a lot of things, and so we can talk about a range of things. But one of the more beautiful areas that you've worked on is saying, okay, if these things are real, we should be able to look at the manifold at time at one time in the same animal, and then a year later or whatever, and find the same manifold structure, but also two different animals, even perhaps two different species of animals performing the same task, if that task is not like some sort of specialized ecologically or ethologically specialized task or whatever. We should be able to see essentially the same manifold, or at least map between them. And so you've done some work mapping between different species and different animals at different same animals at the different times. So could you just discuss a little bit what you did and what you found there?
Yeah, exactly. So this was like work that we did during the pandemic and led by a couple of people in the lab with Matt Perrick. So basically our idea was that there's this common observation that, we can do the same things. And the way usually you do this in talks to wake people up is just ask everyone to close their eyes and touch the tip of their nose and open their eyes, and then everyone has done it, right? So there's two ways that this can happen. Of course, there has to be some similarity in the neural activity, but is it because it's similar brains? But there's two ways that this could go, right? One is because our brains are similar in the organization of, microcircuits, columns, and so forth. And because we believe that this is what shapes manifolds, then the solutions that these brains should adopt should be the same. The alternative hypothesis is that because we know the genome has very limited information capacity, and a lot of people have talked about this, right? Maybe its brain kind of converges into a slightly different solution. So what we did in this study was test the first hypothesis by comparing recordings from monkeys doing reaching and grasping tasks and mice doing reaching and grasping tasks. And we basically showed that these manifolds were similar across different monkeys or different mice, and the degree of similarity depended on how similar their movements were. And we also showed, by the way, that if you try to break the connectivity of the circuits, and we did this in artificial neural network models, to address our hypothesis, right? If you have different underlying connectivity statistics, then you wouldn't get similar matches. So this is how we were trying to close our whole loop of hypothesis from connectivity to manifolds and behavior and then back.
I can use math to do a lot of function transformations, right? And so the way that sort of pictorially you present it is like you can, so you see different trajectories. They look a little bit different between, let's say, two animals, but you can perform a linear transformation on them, and all of a sudden they map onto each other. And so I could skeptically think, I could perform like a fancy transformation on different functions and force them to map onto each other. So what makes you confident that these are the same kinds of manifolds, even though you're doing a little extra math on them?
Yeah, that is like the reviewer number 2 and 3 question.
I'm sorry.
For a few of these papers. So we have lots of controls. One of them, I think, that my friend Matt Perrick, this is also what I brought him up. Like he has done in a different paper from the lab. It's very easy or relatively easy to have RNNs that are very simple models, right? That you train to do the task, and you find different solutions. We also have in the paper the same control. We have lots of controls in papers, but if you compare across two different tasks, the similarity. Let me unpack this a bit more. We have a control in the paper that I think is the most convincing one, where we have two different monkeys doing the same task. Okay. And then we have the same monkey doing two very similar tasks on the same day when we were looking at the same neurons. And these tasks were as similar as I'm generating force with my wrist on different directions, or I'm basically moving my wrist on those same directions. And the manifolds between the two monkeys doing the same task were more similar than the manifolds between the same monkey doing these two very similar tasks when we were looking at the same neurons, because we did this like in blocks. So I think to me that was like the stellar control.
You mentioned reviewer 2, and it just made me wonder like how much these days can't imagine you're getting like pushback on just the concept of a manifold? It seems like there's enough evidence, enough people producing papers, publishing essentially, that any resistance that might have initially been there maybe has gone away. But what do you find? Is there still a lot of resistance to the concept of manifolds, or it's part of the daily vernacular where I am? So
yeah, no, I agree with you. I mean, I remember when I was a kind of junior-ish postdoc talking about these things, you would get a lot of so for context, this was like maybe 2016, 17. There was a lot of argument more about, is this concept useful? Like, will it tell us something that we haven't been able to find at the level of single neurons? And I think now we have enough evidence for that. Now I think that, and maybe maybe a bit too much, because I feel like science should be more like confrontational, in the sense that we have more of these discussions. So I think we should also, now there's not those types of counter arguments. It's more like, how is this not trivial kind of question, like answer. Like, of course you find this, what have we learned? Which is, what a good study should be, right? I think it should teach us something new. But I think I agree with you that the conversations have completely changed in the last 6, 7, 8 years.
yeah, well, I mean, I can personally tell you, like, any which way I look at my data, right? So the manifolds are beautiful, right? And so I get this data set and I think, man, I'm going to find the manifold for this behavior. And then I look at my data and I'm like, oh, it's not like obvious that there are these repeatable trajectories. And then I doubt, like, whether the concept of but you can still say, oh, it's a manifold of X dimensionality. So many dimensions explain so much of the variance if I do some linear PCA dimensionality reduction on it. And you call it a manifold, but it isn't necessarily beautiful. So the question of that you mentioned, how useful are they? One of because they're descriptive and they're obviously beautiful. They're nice to look at. If you can talk about, like, something has a ring shape, or, a linear attractor, a line attractor shape. So these things are nice to look at. And one of the things I wrote down to ask you about is whether relates to their usefulness. Like, what potential do manifolds have in terms of developing theory, which people often point to as being a lacking thing in neuroscience, right? So we have these in-between emergent structures, and we can describe the properties, and they're invariant, but do they help with theorizing about the brain?
Yeah, I think this is what what we as a manifold community, as much of myself that is part of the manifold community,
right?
Is what we
You're
need
like the
to do
leader.
next.
You're one of the leaders of the manifold community.
Man. So I think this is the next phase, right? Like, one thing that is making me happy, in the sense, is like people are studying systems that are a bit different from ours. Because when I was a postdoc, like, you looking at our data, I was like, oh, we're going to find these manifolds, and we find like very beautiful, simple, understandable things. Like, we have all these tasks, and we find dimensions that I don't know, commands to these muscles, and we never found anything like that. But on the other hand, people came studying other systems, like this head direction system, for example, or, we've talked about it to show that, the ring is preserved. In fruit flies, we know that you can drive the activity to different points of the ring attractor that they also have, and you can basically steer the fruit fly, to start to show causality, right? And there's now, like, I think 3-4 papers that we can discuss that show that this compressed description of neural data, also, like, if you take the insights that you can get from them and you causally manipulate in the way that intuitively would make sense, leads to the effect that you would predict. So this is why I meant, like, they are real, but they also are meaningful to the animal, because we can take these compressed objects and basically manipulate behavior by moving activity along this manifold, now that we have better technology. If you want, we can talk about one of these examples. But
yeah, sure. But I also, maybe before that, Earlier when we were talking, I was about causality. I was thinking, well, yes, okay, so the manifold sits in between the nitty-gritty physiological details and the behavioral or cognitive output, but also behavior has an effect on neural activity, and behavior itself is a constraint. So in your example of closing our eyes, touching our nose, it may be that I was going to call that ethologically relevant behavior, although it's not, but a behavior like that we've been sculpted evolutionarily to perform. It's like vital that we be able to touch our noses to survive.
Yeah.
But that could shape the neural dynamics themselves. So how do you think about that causally in a sort of, is there circular causation there or,
Yeah, I mean, like what we saw in the paper too is that if you look at the preparation of these movements while the monkey is not moving, you also find these preserved manifolds. And again, like also like the example during sleep in the head direction system, and also the grid cells that are related to navigation. Like I think this supports the reality of it, of manifolds, but. I think the causal effects have to be a bit more direct, right? And as you said, like as dissociated from behavior as possible, right? Because otherwise we don't know if what we are doing is is the behavior, the manifold is a representation of the behavior or a correlation of the behavior. I didn't say representation, a correlate of the behavior, and then what we are doing is manipulating this, right? And I think there's a good example from maybe we can talk about a concrete example from my now colleague Joe Patton here at Champalimau, who has worked on this. Like he's very interested in timing in the nervous system. So they've developed this task where basically rats have 3 ports and they poke their nose into one port to start a trial. And then they are presented with then a tone, auditory tone rings. And then there's an interval and a second tone rings. And the rats basically have to categorize if the interval between these two sounds was smaller than 1.5 seconds or longer, shorter or longer, sorry. And if it's shorter, they have to stick their nose in one of these goals and of these wells. And if it's longer, they have to stick their nose in another one.
It's an interval timing task.
Exactly, a categorization task. And an important detail about the task is that they have a version where the rats cannot move, so they have to be there like with their nose during the interval. Right? And what they've done is like they've found that basically in the striatum, so in the basal ganglia, they find like a manifold that is like basically a trajectory that is independent of the passage of time. So it's always the same geometry. What happens is that the speed of these dynamics along this manifold relates to the passage of time. Okay, so what they've done that is very cool and causal because the rat was like they're waiting. They could cool down a striatum to make this dynamics go a bit slower, and then the rats would report that less time had passed. So to me, this is like really beautiful because there's, as far as they could see in the paper, no overt correlate of this. The rat was just like internally like misjudging, if you want, based on this internal dynamics, the passage of time on this manipulation. So this, I think, a very compelling example that these compressed objects allow you to make some predictions that then you can test causally to show that they matter for behavior.
They physically cooled down was it basal ganglia?
Yeah. With a temperature probe. They also showed that if you can heat it up a little bit. So the paper, I think, has like 4 different temperature manipulations, and you can see that the speed of the dynamics and the behavior move in the way that you would expect.
And I'm so sorry, and this is kind of a detailed question, but one could imagine that 2 single neurons are mainly responsible for all the variation within that loop structure and speeding up and slowing down. So I guess the question is, how are we confident that they're manipulating the manifold as opposed to a different explanation of manipulating the speed of all the neurons? And so you can explain it by manipulating all the neurons, but I guess that kind of defines the manifold. I'm walking myself in circles here.
Yeah, no, but this is like a very good point. And I was thinking about this recently in the context of, reading about emergence. Like this is a topic that also comes up in your podcast and I'm interested in, right? Because it relates a lot to manifolds. So I think the intuition here, and maybe for everyone, or at least my intuition is that, manifolds can be reduced to neurons, right? Because manifolds reflect the collective activity of all these neurons. But I think that it's a bit like Phil Anderson's More is Different paper. Unless you've observed the manifold, you cannot predict what is going to emerge. So you can go from manifolds to neurons, but you cannot look at the neurons and expect this is going to happen. You have to basically analyze what these neurons are doing as a collective to see the manifold.
Mm.
So I think it's a really beautiful study because it means that, again, this manipulation either makes whichever downstream region judges, like, I think it's been this time, go left and right. That is not what I think happens, or maybe moves the collective brain state to that is related to the decision of left or right.
All right. So let's say you have this in the basal ganglia, you have this beautiful timing manifold structure that you can either go slower or faster on, and you can causally manipulate the rodent's behavior to subjectively or to report its subjective report of the timing. So, but then, all right, so then you have that in one brain area, and it has to causally interact with other manifolds in other brain areas. And this is something that you were alluding to earlier, right? That this is a challenge. But if I take the neurons from that, and then I treat it not as one brain area, but then I take a different neural population from a different brain area that is part of the causal story of how the behavior eventually happens, and I treat them as one brain area, and I get a different manifold from combining the two and their interactions. How do I think about that? I can potentially look at the manifolds of every brain area.
Exactly. I think this is one of the two limitations that I was alluding at the beginning. Like, it's like basically a different side of the same coin, in the sense of, I think thinking about brain regions is very useful, right? Because even if, again, I don't think the brain is like 100% modular, I also don't think the brain is 100% like everything is everywhere all at once. So again, it's something we can talk about. We can say like, oh, prefrontal cortex neurons mostly care about this. If we listen prefrontal cortex, this happens, and these things, right? So in a way, they are useful, compressed understanding, and we have centuries of evidence on these things, right? On the other hand, to what extent the most interesting things that we do, like having this very abstract conversation or, creating art, can be reduced to this area is creating art or making a decision. That is I think the big challenge for going from behavior to this neural implementation. And maybe what we have to do is what you were alluding to. We have to take all the areas together and look at some change in the brain-wide manifold that will be the correlate of this interesting thing that we're interested in, right? Like an interesting behavior.
But then you could just measure the behavior and say that is the manifold of the whole brain, right?
Exactly.
Like what granularity do we need to be satisfied?
Exactly. I think that is the $1 million question. And this is when you asked me at the beginning where I was at personally. I think manifolds, or I'm quite convinced, and I think we've been pushing ourselves to be convinced that manifolds are the ideal level to look at neural function. But I don't know if we will have an understandable mapping from neural function to behavior that works across everything. And understandable, I mean, in this, I think you had like Hank Direct in the podcast, right? The understanding scientific understanding. Hank Direct.
Oh yeah, Hank. Yeah, you cite Hank's work. I've seen.
Yeah, like talking about scientific understanding. Maybe we can build a transformer that will be able to one day build this mapping, but maybe we will not be able to understand it. Right?
We can't talk about it.
I'm interested in understanding it.
Yeah, I'm interested in understanding too. And in some sense, we're limited or trapped by these symbolic things that we use called words. And we have this legacy of what's known as the machine metaphor for brain function, where we have to look at like a little part and give a name to a function. But everything that we're learning is that it's the name that we give to it can change depending on the context of what the task is that the animal's performing. And everything is dynamic and changing. And we just can't have a conversation about we can't go to like some differentiable limit of function. We have to like give it a static name at some point. And we can't like say the hippocampus is a navigation memory system when you're waking up in the morning, when you're 42 years old, and when you're 47 years old, when you're going to bed at night, it's a abstract, you just get into an infinite run-on sentence. So this is part of the challenge, I think, is that we have to give names to things, basically.
Exactly. If we want to understand them, right? Like, although language and thought are not the same thing, right? Like, at least we have to have, like, again, like compressed objects we can think with, right, to understand. And maybe behavior is the best level for that.
Maybe behavior is the
maybe behavior, as you were alluding to, is the best level for that.
Wait, so then now I've lost again where your thought is in terms of how you're thinking about these things. So are you with someone like John Krakauer, who says like, well, there's just going to be a divide between the psychological and like neural descriptions, and we just have to get comfortable with it?
I think I'm not in the same extreme as John, for sure. But I'm not convinced that we would be able to find a neural explanation of all the interesting things. I think we have good neural explanations for interesting processes, right? Like these things we talked about, constraints, or also like what is the transition between preparing and executing a movement, or this time judgment decisions and things like that. But I don't know if we would be able to, by the time that you and I retire, we would have cracked this.
Not me. Yeah.
I think you are a bit farther away from retirement.
One of us, at least, it probably is.
Yeah.
So,
so I'm optimistic just to clarify, I'm optimistic as manifolds are the best way of neural description, but I'm not 100% confident that we'll be able to find a neural explanation of behavior.
Let's talk about causality for a second. Going back to it, I know that you just got Alicia Huarero's book, Context Changes Everything. And I have so she's been on the show, and I think I mentioned this to you, that some of the feedback I've gotten from her book, a lot of people really love it. I really love it. But I also sympathize with some of the neuroscientists' feedback that it's kind of like, well, okay, fine, but I don't know really what to do with this information. I can't put it into equations. There's not some formal mathematical system by which we can study how the constraints of a system, how to think about how constraints affect ongoing behavior, whether you're talking about behavior of neural populations or behavior of the organism. But one of the driving points is that, and philosophers have written about this, is that there's this hazy line between what we think of as causality, like one billiard ball hitting another, and the constraints, namely the pool table and the composition of the wood of the cue, etc. All of those constraints that go into making it happen as well. And I've just come to sort of accept that the billiard ball view of causation, let me rephrase that. I've come to accept that context and constraints are the main story of causation, not some side show of causation. And I'm wondering how you think about, if you thought about these sorts of things, and if so, how you think about them, since we were talking about causation.
Yeah, I haven't read Alicia's book yet because I just got it a couple of days ago, but I really like I define manifolds as mathematical objects that reflect real constraints on neurons, right? So this is something that I'm very interested in, and I think it has a lot of power, right? Like
But
in
see, even
neuroscience.
in that wording, sorry to interrupt, but in that wording, it's a mathematical object that reflects the constraints. So
Exactly.
the constraints themselves are sort of like, they're there, but it's really the manifold is the thing, you know?
But I think the manifold is, if you want, a consequence of the constraints, right? Because if we didn't, maybe I was being a bit wishy-washy with my wording, as we have said, like reflects those constraints in the sense that, or is a consequence of these constraints. Because if neurons, if we didn't have like connectivity structure that improves constraints and we didn't have common inputs, neuromodulators and this stuff, we wouldn't have constraints, right? So that, to me, is what delineates a lot of what a neural circuit can do. And if we think, for example, about learning, we talked about this paper in monkeys where the constraints that you have shape how easy it is to learn a slightly new version of what you know how to do. So I think constraints are very important and have a lot of causal power. The billiard ball causation example is really nice because it's, again, easy to understand. And the example I gave you from this time manipulation study is very, very billiard bally, right? You manipulate, and this is the consequence. But I think the other point about constraints having causal power or on what animals and brains can do is like super important. And clearly it has been like the basis for many of the studies we've done in the lab.
One thing I also want to ask, and I want to make sure I ask you about this in the manifold story as well, because we have other things to talk about here. But everything that we've been discussing is about measuring the spiking of neurons and correlating the spikes of one neuron with the rest of the population of the other neurons. But then even in your last few sentences there, you talked about, I think you just mentioned the word neuromodulators. There's a lot of other things going on in the brain. And we tend to think of the spikes as being the thing. The spikes are, after all, what make up the manifold. The spikes are what we measure. But there are neuromodulators that shape the activity of the spiking activity. But are we even measuring the right thing? Like, is the spike the right thing? How do you think of? I know this is like such a dumb question, but we sometimes have to go back to those like assumptions, like because of that, it's the lamp post problem, right? So we could measure neurons with single electrodes a while back. And so we did, and we averaged them, etc. Now we can measure the spiking activity of lots of populations of neurons. So we are, and we have found that they have manifold kinds of structures. But is this the same sort of problem of looking under the lamppost just because that's what we can measure?
Yeah, that's a great question. I think that I also wonder about this, but I think, and we discussed it a bit in the review that we were talking about. I think, for example, neuromodulation will change manifolds, and in a way that looking at, of course, if we had measured them, it would be better because then we could basically hopefully establish relationships between the geometry of the manifold or where you are with respect to, I don't know, norepinephrine release, right? But I think this is kind of built or captured by the manifold. The problem is that if you don't think about it, then you may be seeing something on your data that is like, the animal is doing exactly the same thing as far as I can tell. Why is the activity on this part of a stereo space and not there? And maybe it's just like this, if you want, neuromodulatory states, or maybe like arousal or other things that are also related to neuromodulators, right? So I think it will be reflected in the manifolds. Not thinking about it may add to our confusion, or has added to our confusion, I would think. And there's a couple of papers that hint in that direction. Yeah, and the more we can measure, the merrier.
Sure, the more the merrier. But I guess buried in my long-winded sort of comments or question was, would it be better if we defined manifolds, if, like, let's say neuromodulators were part of the manifold rather than shaping the manifold? The manifold is the neural activity, but why can't the manifold be neural activity and neuromodulatory activity and oscillations? There's this thing is like, that's the grand goal, right? Is to, like, we have all of these interacting parts. that we know give rise to emergent properties. Can we tell a story across temporal and spatial scales, including all these things, not just spiking, and call that some sort of manifold? Would that be better? I don't know.
Yeah, I don't know either, because the neuromodulators, I'm sure that, I'm sure there's a couple of studies that show that, for example, there's correlated drift, quote unquote drift, for people that are not watching the YouTube recording.
You have to define drift. Define
Yeah.
drift.
Or some unknown slow change in neural activity that was found across very distant brain areas that is basically related to engagement and attention, that could just be explained by, there's this change in neuromodulator release that basically makes activity move in the state space, or drift, if you want, in those two areas in a correlated way, and that just may be arousal. So if this group had been measuring like this neuromodulator, they could know for sure. Going back, you talked about neuromodulators and oscillations. So this is something that we actually worked on a small paper and looking at the relationship between manifolds and intracortical local field potentials. And we found a relationship that was kind of stable between different phases of behavior. But one thing that I'm not sure about is whether oscillations are like causal for the brain, or if they just reflect underlying biophysical processes that with neuroanatomy just give rise to these oscillations that we measure, and they're just like epiphenomenal in that sense. And I don't have a strong view on this.
You don't? I'll argue for the fact that they're causal, right?
Okay.
So let's say oscillations, you have a bunch of neurons, and then you can measure the field potential activity, right? So you have like sort of positive, negative, positive, negative. Well, neural activity is affected by the voltage difference between the extracellular milieu and intracellularly. And if the oscillation, which is the collective whole, is changing that neural activity that the single neuron is being affected by, how is that not causal?
Yeah, I think it could be. I just don't. What I meant in the most general sense, because there's a few papers that I'm aware of where they saw that the biophysics of LFP generation, for example, depend a lot on the brain area that you're looking at. Like, for example, hippocampus has a very different structure than motor cortex. So there may be like the sources generating the oscillations that you measure in these areas may be local in some areas and may be not local in other areas.
Mm-hmm.
So it just meant like using this idea as a blanket, like basically a blanket assumption that they are always causal. And this is, I think, like a different, conversation. I think like to be, really well informed, I would have to study like these mountains of papers that I was trying to read back then.
Well, we'll let Earl Miller do that, right? Is that the...
Yeah, exactly. and Bijan Pesara also. He has some interesting causal evidence that specific band, if you modulate it, can have an effect on behavior. So.
Well, I think this is, again, the problem is that there are so many interacting parts. And you guys write about this in your review too, is like, it really is dependent on what temporal scale you're looking at, on what spatial scale. And that's why, going back to like, the degree of modularity of the brain, whether area X is always performing function Y. No, it's not, but it always is in this one condition. So how can we describe what area X is doing? I mean, I think that same thing can be said about oscillations and astrocyte contribution and all these interacting parts.
Exactly. Yeah, astrocytes are also like things that we should probably talk about as a community that have been like, well, there's people studying astrocytes, right? But maybe they don't get astrocytes enough credit.
Right. The astrocyte manifold hypothesis.
Exactly.
You and collaborators have written about how, like, especially in the early days of figuring out what a manifold was, you take like a high dimensional space, like, lots and lots of neurons, which means lots and lots of dimensions, and then you throw it into like a linear dimensionality reduction technique, most famously principal component analysis, and then which shows you like the linear shared variance between the neurons. And then you have like these little well, you can map out a manifold in that linear space. One of the things that I guess you've worried about over the years, or continue to worry about, is is that too simplified a picture? Because neurons themselves behave in nonlinear ways. Even dendrites of single neurons can behave in nonlinear ways. Then why would we expect to get this linear relationship in a low-dimensional manifold? And what you suggest is that, well, yeah, that probably is too simple. And it turns out manifolds are nonlinear, and there's work to do in mapping out the relation between the complexity of tasks and cognition and the nonlinearity of manifolds. So can you just discuss briefly where your thoughts are on that?
Yeah, it's like like from the beginning, we were thinking about this because it would be so nice, right? Like that all things are flat for some reason, even if nonlinear neurons, as you were saying, are nonlinear, and if you have like many nonlinear interacting parts in complex ways, it's unlikely to be all linear, right? But on the other hand, like PCA is nice because, as you were saying, principal component analysis is the simplest method, most basic assumptions that you're basically asking, what are the patterns within this population that explain the most stuff? And you could make the inference that what dominates this population is likeliest to capture its function. So I'm, very happy with starting with PCA. But then we're starting to look at more complicated tasks where, we needed lots of these principal components to capture the variance well. And what we found is like what you would expect that you were describing, right? If you have like your cereal bowl, this is a two-dimensional surface, but, it lives in a three-dimensional space. So with PCA, you would need like 3 dimensions to describe this bowl. And things get even worse because, our world, unless you are a string physicist, has three spatial dimensions. But you were saying, like, we record now from hundreds or thousands of neurons. If you think about we go back to the ring manifold that we were talking about before, that a ring is clearly a one-dimensional thing, right? It's like a ring. But if you take, you can think about it. I wish I had like some rope or something here. Like I have a rubber band. So for people on YouTube, if I take this rubber band and this is a one-dimensional manifold, but if I twist it a lot, now it becomes a 3-dimensional manifold. If you look at how many linear dimensions you need, and if you have 1,000 neurons, then it can look like a 250-dimensional manifold, even if it's just a ring. So I think there's been a lot of papers written on things are very nonlinear, things to scale with more neurons, but we have to be very mindful about this simple example of the rubber band, that you can use a nonlinear dimensionality reduction method and probably find a low-dimensional surface. And I think this matters just because of the assumptions that we were talking about at the beginning, right? Like there's constraints and that are intrinsic to the circuit and the task itself.
Yeah. But part of Henk de Recht's concept of understanding is like that we should be able to like make sort of predictions without doing calculations. If we understand something, someone who is well versed in, let's say, manifolds should be able to sort of like look at the shape of a manifold and make a prediction about the outcome of some causal manipulation or something. I don't know what Henk would actually say. I think he would say that. But so in the case of like, okay, we do this linear manifold reduction technique and, you could see it. If you do like a little smoothing technique, you can just see that like nice trajectory. But then you do the nonlinear one, which explains more variance, and is epistemically a better explanation for the data, but all of a sudden it is sort of escapes our intuitive ability to think about it and manipulate it. Where's our satisfaction there? Like, where do we cut off where we say we're satisfied? Is we've estimated it well enough with linear, let's be or like, yeah, it's really nonlinear, but if you look at it this way, you can see like a little glimpse of it in the linear domain or something like that.
Exactly. I'm with you. I'm more interested in understanding. So like the task that we looked at, like the nonlinear manifold looked like a slightly different version of the nonlinear.
Right.
Just like we needed fewer dimensions. So this would be interesting when we as a community to record from people playing Tetris or, creating music, right? But to me, it would be more satisfying what you were saying, right? Like we look at something that we can comprehend and then that we can use to make predictions and test hypotheses. Why I think this is important to bear in mind is to avoid saying like manifolds are not true because I found that the more neurons I record, the more dimensions I need with PCA, with principal component analysis. And this is a bit technical, sorry, apologies to the audience, but I think this is what I think is important.
Okay. So that's what I was going to ask is like what kind of purchase that has on whether we consider manifolds ontologically real or just real. I didn't have to say ontologically. They're being real if like it escapes our low dimensional understanding of them. And we just have to like sort of trust, well, it's this really nonlinear nonlinear space that we can't conceive of, but we just have to trust it. But it's still real.
I think we should be able to even if it's non-linear, like a lot of these methods, basically like the one we use, TiSOMAP, you can think about it as you have a very non-linear surface and it basically unwraps the surface, right? An object, like you have your, I don't know, your cereal bowl again and it will unwrap it. So I think you just have to adjust your thinking. And, there's mathematicians that think in 10D. I think like everything in science is a compromise between like, and we can go back to Henk Direct, making predictions that maybe are not 100% accurate, but I can make just by reasoning about them, and they would be like 70% accurate, versus I'm going to build a huge neural foundation model that will give me 100% prediction, but then the model is so big that I cannot just like make sense of it. So I think it's like the art of modeling and understanding. I think this is why science is a bit like an art, and hopefully we still keep our jobs with the current AI revolution.
Well, I mean, Masrita Chiramuta makes this point too, and this is an old philosophical point that, when we model, you just said modeling, it's necessarily an abstraction. And going back to Alfred North Whitehead, which he called the fallacy of misplaced concreteness, one of the things that we tend to easily do is slip into treating the model as the real thing. Right. And so there's this sort of worry that when we're talking abstractions and models and manifolds, that we can start treating the manifold as the real thing when maybe we're just modeling it as this something because we have to be able to do that. So I don't know. There's this trade-off always in thinking about these things.
Yeah, I think this is one thing that used to come up a lot in the early days about talking about manifolds. But like, is this abstraction real? But I think, I wonder what would have been the case if we had been able to, if the history of neuroscience had been different, if we hadn't grown into looking at single neurons, if it had been the opposite, right? Because, like chairs are not real, right? They are collections of atoms. So maybe we had seen atoms before, we would have said that chairs are not real.
Oh, man. Okay. Yeah. I've asked David Krakauer, like, is there anything that is not an emergent property? And he said, yes. I still don't buy it. I think everything that we can talk about is an emergent property of other things.
I guess like the quantum wave function, if you asked Sean Carroll, right, that is everything is an emergent property of the quantum wave function. And I think I may agree with that to my poor understanding of quantum mechanics.
Right.
But anyway, like thinking about the quantum wave function will not let us predict how our conversation is going to go, right?
Yeah. In a sense, though, like, I think you said, did you say a neuron is not real, or a cell is not real?
I said that if we could only look at atoms, we may say that my chair is not real. No, neurons are real.
Yeah. But we could kind of say that like a neuron's not real because it's only atoms, right? It's just where it pans out in a super reductionistic.
Exactly.
I think it's funny that we always use atoms still, even though we know there are quarks, subatomic things, but we still go to atoms as the base layer,
like baryonic matter and these things.
Yeah.
But I guess the idea of atoms is the oldest one, right? Like it's in the Greek.
That's right. Yeah. Democritus. Yeah, I can't remember
I think so.
atom.
I think so.
Yeah. A neuron, so manifolds are in this interesting space too, because a neuron, even though you say it's not real, Juan, I'm just kidding. But you can
I hope
see
this
it,
is the opening.
right?
Yeah.
Neurons aren't real will be the title of the of this episode. But I mean, you can see a neuron and you can measure like the
Yeah.
voltage, right? And that's how we get action potentials, is by measuring those voltages. So manifolds, there's this like, they're like these floating abstract things, right? So there's always this where I can't see a manifold unless I plot it like in a three of its dimensions, right? So
Yeah. No, I
there's always,
agree with that. It's like a good, you know, thing to say. Like, it's obviously right, but I think we have enough evidence that that is the case, right?
okay. I've
I think.
heard a lot of people say, like, when they talk about you, they say his only talent is manifolds. That's all like he can do, or he's just jabbers on about manifolds all day.
Well,
So I'm going to try to get you to talk about something else today. But I know that I don't know how you maybe we can transition into your work with BCIs, and because I was going to ask you in the very beginning, like, how you got into this racket. Are you still doing these days what you are you following? Are your interests the same these days as what got you interested in neuroscience, for example, in the beginnings?
yeah, so I've had like, I mean, this could go on very long, like my like the journey of my interest. Like, I started doing research a bit in robotics as an engineer, and then I moved into more translation. I think what people would call now neural engineering. So my PhD thesis was on building a closed loop system to use surface stimulation of muscles. So for people who are old enough to remember these ads of machines to build up your abs while you were like eating a snack watching a football game. So we would use like sophisticated ways to stimulate forearm muscles and arm muscles to counteract the tremor of people with Parkinson's disease or essential tremor. So this was my PhD, but at the same time, I got very interested in understanding why people had tremors. So I started to read a lot about clinical neurophysiology. I went to work in a lab recording from human spinal cord motor neurons in people with tremors. So this was my segue into neuroscience. And then I guess because I had this like translational interest, this is why I joined like Lee Miller's lab that, was like doing this pioneering work on, for people who don't know Lee, like he was one of his group, was one of the first, like my friends Emily Obi and Chris Fetier did the work. As PIs, we don't do anything, right? They showed that a monkey that they had like basically reversibly paralyzed with anesthetics, like the same anesthetics that the dentists use. So they were paralyzing the nerves and the monkey couldn't close the hand. Like they could decode from the brain using a brain computer interface how the monkey wanted to activate the muscles. And then they could use the same type of muscle stimulation technology I was using to reanimate the paralyzed muscles and let the monkey do interesting things again, like grab things and move them around. So this is the project I joined, and I was working quite a lot on this. We only never had an n equals 2 experiment. So for people doing animal research, they would understand this, like the implants and things. So I was working a lot on BCIs, and at the same time, again, because I was interested in understanding what was going on, this is how I got into Manifolds during my postdoc.
Ah, okay.
So I was kind of doing both things at the same time.
So I was thinking, like, I didn't know if this was a sort of a newer direction, but it's like returning home to your original interests.
Yeah, and in London, so when we started my group, like we were doing this manifold work. We are also doing a lot of mouse experiments that we are presenting at conferences now, more like motor control, motor learning, BCIs. And we were also working on motor neuron control because we are a motor control lab at heart, for those who don't know it. So we mostly work on motor control and motor learning, and the output of basically movement doesn't happen because the hand magically moves, right? Like your motor neurons are doing causing the movement, right? So we are studying that and also translational applications. And this was a bit of a long answer, but just to put things into context.
Yeah. So spinal cord injury. So tell us the story about what you have found in patients who have spinal cord injuries and the extent of the damage versus what you can do with it, etc.
Yeah, so I'm very excited about this work that we reprinted recently, because as part of all this like basic understanding how spinal motor neurons work efforts, like a bunch of awesome people working in the lab. So just like shout out to Vish, Kira, Xingchen, Agnese, and Lara. So it's like this was a team project with my colleague Dario Faina. So we were basically doing all this basic science work, and then we had the idea, why not basically try to have even clinically complete spinal cord injury people control their motor units, so their motor neurons, to do interesting things. Like, in other words, can we build a BCI that is just like putting a sticky electrodes on the arm of a paralyzed person, and what can they do? So this
you said clinically complete.
Exactly.
What is that? Incomplete in another language?
Yeah, this is a very thanks for asking, because clinically complete means that they basically cannot generate overt movements, and and if they are sensory complete, they cannot have they don't have sensation. But a complete spinal cord injury would just mean you take a chainsaw and you cut the spinal cord in half, which people wouldn't survive something like that. So this is very important. Like even clinically complete people still have some residual input from the brain below the injury. So this is what we were hoping for, that we can put these sticky electrodes, use our math tricks to identify motor neurons, and that they would be able to control these motor neurons to do tasks and play games, basically.
Just before you get into the details of that, so just what is the range? So clinically complete sounds like one thing, but there's got to be Bill has 30% of the projection neurons still somewhat intact, and Nancy has 45%, but they're both clinically complete or something. So is there a typical range at which there's still some activity going down, or is it just continuous, or what?
Yeah, I don't know if we have a good quantification of that. This is usually done, people do imaging, but this is usually done with clinical rating scales. And also what we were doing was using ultrasound of the muscle. So we would put ultrasound probe in the muscle and ask participants to try to contract different muscles. And in the clinically complete participants, we couldn't see fibers contracting most muscles. So that is the extent of it. So there's the
you don't necessarily even have a measurement of how extent the damage is through the trajectory. Okay, yeah,
exactly. We don't, and there's a lot of there's a lot of interesting translational work by many people like Marco Capogrosso and Reward Curtin and others, where they show that you can do a lot with these spur connections to basically retrain with neurotechnology and have people walk again or use their hand again with a neuroprosthetic. But we want it to be very low tech. It's what if we put sticky electrodes, and can you just use your computer to communicate, to play games? Can you navigate your wheelchair with doing this, even if you cannot move your hand, right? And what we found is that they could control up to 3 degrees of freedom is how far we got. We are continuing these experiments. So they could, for example, one of them who couldn't move his own wheelchair, we didn't control the wheelchair directly. This was a virtual wheelchair because, the engineering time.
But it could be, in principle, easily used.
It was. Yeah. So it was basically turn left, turn right, go straight. And he could do this with motor neurons from his paralyzed hand. And for him, he was oh, this is great, because I will be able to move around my house on my own.
Yeah, God, yeah.
And I'm also I was also working on BCS, like you were saying, but some of these participants were open to having implants, others not, because they've already gone through a lot, right? So I think having this big range of technological solutions is exciting. Sorry.
Oh no, I'm sorry. Would that not be considered BCI? Using the EMG to control? I'm not sure what constitutes the computer part of the BCI then.
Yeah, it's I was actually looking at the brain computer Interface Society released this definition that is a paragraph long, and maybe it would fall within the BCI because I can't remember if it actually talks about interfacing with the nervous system. But I'm just trying to be conservative, and we call it motor neuron interface.
Well, it's because the B is lacking, not the C in this case. Yeah.
Yeah. But yeah, I think this is cool because now we're interested in pushing this forward as a new way to, assistive devices, but we're also thinking about applications for rehabilitation with these approaches.
And how would that happen? How would the rehabilitation happen? You can train the regeneration of the nerves?
Maybe that is the question. Like, maybe we can actually help, because rehabilitation is basically you need like intensive training, right? It's correlated with having better recovery. And
yeah,
our mutual friend John has like a lot of work showing that, right? Krakauer. So maybe the problem if you are fully paralyzed at the beginning after you have a neurological disease, and we don't have to think only about a spinal cord injury, like you cannot exercise, right? Because you don't get feedback about what you are doing. But if you are using this technology as a way to basically close the loop, because you are controlling even like this motor neuron that you can capture, then you could have people retrain their nervous system. Of course, this is...
How's that more effective than or different than just behavioral rehab, which should in principle maybe be doing the same? That's what people are trying to do when they're doing behavioral rehabilitation, right?
Exactly. But the problem is when you cannot move at all, right? You don't get any feedback about what you are trying to do. Like you are not getting...
you don't know if the thing that you're...
Exactly.
Sorry. Yeah.
So you don't know what you are trying to do because you cannot practice this because you don't see what you are doing in a way.
You can't correct any errors because you're not making any errors because you can't see what you're doing at all.
Exactly.
Yeah.
That is what I was trying to say.
Okay. Yeah. So you're getting the feedback, and then it's the error corrective mechanism that should in principle reinforce, and then there's some plasticity involved, etc. That
I think it's also like the simpler thing, like observation that if you have people play, like stroke participants play with video games with exoskeletons for many hours a day, they get better than if they're just doing 1 hour of physical therapy. So it's about like gamified interventions that people are engaged and practice many hours a day. So there's
they're willing. They're like willing
exactly.
to do it because it's like more fun, and that's okay. Yeah.
Exactly. And you are getting this, like in a way you are engaging the system, and you're also getting them to practice. So I think it's both of those dimensions. But I'm not an expert in clinical neurology.
Right.
But I think that is our prediction. That is something that we are very excited to build here in Lisbon, like we're working on actively.
Well, you had mentioned, is this part of something you had mentioned to me, which was like a big clinical and translational neuroscience push where you are?
Yeah, I guess like it is. So I just moved to Lisbon to join the San Palimo Foundation. That is, I think, like neuroscientists know it because of the like the very preeminent, basically, if you want neuroscience program, right?
Congrats, by the way. Congrats on the move.
Thank you. And now so I joined that, and basically this big expansion into translational neuroscience. So there's this new Center for Restorative Neurotechnology, directed by John Krakauer, whom we've mentioned like a lot of times. I guess he's your VIP in the podcast, so he's
he's a frequent guest, we'll say. He's a frequent visitor of the podcast, friend of the show. Exactly. Yes, for sure. Yeah.
So I joined this effort, and this Center for Restorative Neurotech has 3 legs. So one is this translational neuroscience program that I'm part of, that we call Neuroscience of Disease. Then there's also like a clinic where a lot of my colleagues here are actually clinicians, as well as like neuroscientists. So it's a lot of fun because I'm in meetings. I learn a lot about psychiatry and neurology, where we will be administering like neurotech and AI-based gave me 5 therapies. So I mean not me, but my colleagues and people in our group. And then we have a warehouse that is literally a warehouse that people who came to Costa have partied at a few years ago. And this is now basically, and we have a lot of like groups doing like hardware, software, tech, and we are starting to have like some startups too. So the idea is to go from basic neuroscience to clinical trials and translation in the same building by the Tejo River here in this one. It's exciting.
So where does this fit in your wanting to understand versus wanting to engineer things?
I think it's like, it kind of seemed like a perfect fit because the lab was working on both, right? On understanding and bringing what we're understanding into, fixing. So in a way, it's like the perfect space for us.
What have we not discussed that you're finding exciting in what you're doing these days?
So many things. I think we've covered most of the bases. The other things that we're working on is basically mouse experiments to try to understand closed loop motor control that build a bit on these ideas that you mentioned on the email about, like different parts of the brain working together and there not being like a clear one-to-one mapping between function or what a brain area does. So we are doing experiments along those dimensions in running mice. We're also doing some BCI Batista-inspired neural constraint studies, but I'll let you jump in.
Batista-inspired. Well, that's what we were talking earlier about, sort of the difficulty because we're confined to use language, right? And what we use, the difficulty in thinking about going beyond a hyper modular, very static view of the functions of different parts of the brain, et cetera. But part of the problem is that we write scientific papers using language. How much of that is like part of the problem is just writing down sentences without trapping yourself into that sort of old view of like the static, I don't know, it's kind of a reductionist small view of it.
Yeah, I think part of it is like we should make an effort to integrate different approaches, right? So look at different tasks, for example, maybe different species, so that we also have to be mindful differences of species, right? Like, like you and I have worked in mice and monkeys. Clearly, mice are not small monkeys and we are not big monkeys.
Yeah. Monkeys have better neurons, by the way, better neural activity in monkeys for those listening.
Yeah. Exactly. Yeah. So there's this integrating across different tasks, looking at different methods like correlational, looking at activity decoding during interesting task designs may not be enough. We need to perturb, but we also have to be mindful of, are we perturbing just like doing a little poke transiently or selectively versus are we listening? When are we listening? Are we listening before an animal learns the task or after? I think there's these two axes that we wrote like a short opinion paper about. This was with Josh Dalman, and it was basically led by Jimmy Gemas, a postdoc in the lab, and Jason Keller, a postdoc in Josh's lab. We were talking about this, about the need of looking across different tasks and different ways of looking at data. And if you do that, basically you realize that the brain is not isomorph, is not like, a blob. But there's also not clear isomorphism in motor control. And I would argue that if there's not a super clear isomorphism in motor control at the level that we're describing, maybe there won't be for cognition. And then the other need of looking at basically where the animal is with respect to the behavior or its own life. And what we meant by that is like a developing animal is not the same as an adult animal, or an animal or a human, for what it's worth, that is learning to do something for the first time. Like the brain is not the same as when you are very skilled at it. Even if if you look at me trying to play a complicated jazz chord in the guitar or Pat Metheny playing it, I'm sure my motor cortex doesn't look like Pat's. So yeah, like basically looking at all these things and just like trying to stop and think about it, digest the literature, and be mindful of what we may be missing.
It's just D major, C major, G major, Juan. That's all you need, man. That's all that's really required.
Yeah,
My fingers can
that's true.
find those. Yeah.
And maybe mine still can.
Well, you're discussing, was it current opinion in neurobiology is the work,
Yeah.
the nice review that you're discussing, and the challenge of looking across timescales relating to like everything that we've been discussing about how to think about these things and write about these things.
Yeah, exactly.
I think in the very beginning we were talking about like the legacy of like recording single neurons, and a lot of the legacy of neuroscience is also, there used to be, I don't know if it was dominated by there, but in the early single neuron days, it was a lot of it was non-human primates, was monkeys, and that's where I cut my teeth as a neuroscientist. And I just said monkey neurons are better than mouse neurons, right? But it really is like, if you can record the single neuron in a monkey, and in real time, you can listen to the modulation of that neuron and think, man, that is involved in that decision, in that eye movement or whatever. And then you go and you do the same in, like, for example, a mouse, and it's just not nearly as clean. And you think, oh, I don't know, the single neuron couldn't be doing this. Like, it has to be a manifold. It has to be a population. And the neural responses are like so sluggish and slow compared to those in a monkey. It just seems like, how does the brain even do this? It seems so noisy and slow and stuff. So I don't know if like recording also in more recordings in rodents and organisms with brains, or even thinking down to C. elegans with very few neurons, no action potentials as we know them when we typically talk of action potential. And yet, I don't know, do C. elegans, do they have manifolds?
I love this question, because there's this paper that I really like that I think doesn't get as much love as it deserves. And I'm going to butcher the authors. The first author is Brennan, and the second one is Project. And my weird face for people on YouTube is because the last name is like Proekt. So I don't know how to pronounce it. Sorry if he's listening to the podcast. But
It sounds European, quite European, right?
yeah, you would guess like I would have been exposed to that.
Correct. I don't know.
Yeah, probably Proekt. So they took worms. That is like, again, we've known like they have 302 neurons, I think, and they have names. And they took the same worms, genetically identical worms in the same environment, and they could record from the same neurons in these worms, right? Because they have names, the neurons and the worms. And what they did was, the first figure in this paper, one of the first, is like, okay, I have these 20 neurons in this worm, and I can decode the behavior, like, it's turning left, right, or, little worm things. And now I am going to take this decoder, and I will take this genetically identical worm. And what happens? The decoder fails completely. But within worm, they could always decode from this given set of neurons, but they couldn't across worms. Then they did some clever dimensionality reduction to find a manifold. And guess what? The manifold looks the same across worms. The same topology and the same relationship to behavior. So they color coded these manifold shapes that look like a bit like a pretzel that you have bent a bit. And the colors that indicated the behavior were the same across worms. So this is one of my favorite examples with Eve Marder's example that you mentioned, Eve, and that basically you can have basically the same lower level constituents, because in Eve Marder's work in crabs, they also know this is the 3 neurons, right?
Yeah, can you just actually unfold that? And sorry to ask you to do it, but describe Eve Marder's very popular work.
Okay, let's stop with the worm and we switch to Eve Marder's work. Okay, so they've been studying the digestive system, the stomatogastric system of lobsters and crabs. And they have a paper I really love that I can't remember now if it's about lobsters or crabs. But basically they have these 3 neurons that describe what has long been called a central pattern generator. So you can think about it as an oscillator, what Paul was saying, a ring attract. Like the activity loops around in circles. And what Eve has shown is like, you can find this circuit in any in any crab or lobster, sorry. But if you look at the biophysical properties of these neurons and their connections, they are different across animals. And it's not like, you can predict, oh, this is going to be within range of this one. There seem to be like privileged combinations of these properties that give you an oscillator and others that don't. Okay.
And that allow them to digest.
Exactly. So this is another paper that I would like to say that manifolds are ontologically real and have a causal influence and are meaningful because the crab doesn't care about the details of the circuit. It also cares about being able to digest its food.
Oh, right. Yeah.
And again, this manifold is made of these 3 neurons, but there's nothing that they could find in their paper that would be like, oh, these neurons are digestive neurons and these aren't. Like basically it's about the interactions of these 3 neurons and their properties that make the digestive manifold, if you want. So it's a bit the same in the worm. Like the neurons are the same, probably the connections are not the same. But it's still like the emergent behavior at the manifold level that relates to how these worms navigate in the world is the same. This is like maybe a good point to end about manifolds as being ontologically real and having meaning, being meaningful not only to us, right, as people who look at them as like, oh, this is a cute shape that I could put little beautiful colors on, but also be meaningful to the animal.
That's the other possible title for this episode is Manifolds. They're real, damn it. Is the yeah. But I mean, it's interesting to think from an evolutionary perspective and just from a computational perspective, from a what is possible perspective, that you could have lots and lots of different combinations of things that end up doing the same functional thing. Like it's crazy to think that the space of possibilities is almost infinite. The range of what actually works is still really high, apparently. And so you have to think about like these constraints at all levels and like what really does whittle it down to what's usable and what's not. And how do we manage to think about that? And just theoretically, how can we predict, like, can you go the other way and say like, all right, here are your 4 neurons and you have 5 ion channels and 12 neuromodulators. Tell me what's possible and what's not to perform, I don't know, gestation, or whatever.
Yeah. I think this is a fascinating question, right? Like, because the genome, again, has so little information. Like, it's amazing. Like, you're basically squeezing a human like us into a genome, and then you get people that can have these conversations. And I don't know, like, we could play guitar together, right? And at the same time, while there's so much variability in the substrate, it seems so much, but there's still like a lot of consistency, right? Because circuit motifs and columns and layers and areas are preserved, right? So I think this is a fascinating question. And, I would love to have a neurodevelopmental friend around here to basically work on these questions directly. But what you said, there's a really cool paper by Bing Branton and John Tarhill. I don't know if you've seen it, where they basically take the entire connectome of the fly, right? So they have like 2, I think there's more than 2 now, there's maybe 4. And they all have cute names like Fancy and Mancy. And what they do is like they look for a central pattern generator in the connectome. So they basically build a model. And I'll describe it because I think it ties to this idea of manifolds. Like they have this connectivity, right? This connectome, this connectivity matrix, and they know which connections are excitatory or inhibitory, and more or less they can guesstimate the weights. So with this, what they did was like, okay, let's ping this neural network that I've fitted to the connectome and look at which neurons kind of would make the downstream motor neurons that they also have oscillate. And with this, they pick like their 2 favorite candidate input neurons to this like network. And then they just like simulated, then they just like with this neuron, they simulated what would happen to this network that again was fitted on the actual connectome after they were getting rid of neurons. And they will get rid of a neuron and nothing happens. Another, nothing happens. Until they found that just 3 neurons, again, maybe that is the magic number.
Yeah.
These 3 neurons are necessary for our model to make the model oscillate. But the cool thing is that this is a fly, right? So they could be like, okay, now I'm going to go and stimulate this neuron, and basically it fitted the prediction of the model. Like behaviorally, like I can remember the details of that, but it's like it would make it move or not move and things like that. So again, you have all this, but maybe by thinking about what the collective should be doing, that is like generating an oscillation, you can, at the moment, just like computationally find the substrate that you will need for that. So I think when I saw that at a conference, it completely blew my mind.
And they're real, damn it.
Exactly. I should have said that. Sorry. It's late here, so.
Yeah. Well, okay. It is late there. And I appreciate you spending time with me. So the manifolds loom large.
We can continue.
Yeah. No, that'd be great. I mean, the concept of manifold is mentioned so frequently on this podcast. So, but it's almost mentioned sometimes in passing. So I really appreciate you going like deep into it and covering like all things manifold here, and as well as getting to know a little bit more about the other facets of your work. And I'm hoping, and I'm demanding that our paths cross in real life soon, because we do have some overlapping research interests and stuff. So I hope to visit your world soon, and anyway, hope that we can meet in person soon. So anyway, thanks, Juan, for coming on.
Yeah. We should continue this with some beverages.
Oh, very good. Yeah. When I asked you to come back on to chat for a few minutes or whatever, and I mentioned, I want to ask you, what if there's no manifold? You said, well, this kind of overlaps or touches on some things that you wanted to highlight from that current opinions paper. What was that you wanted to highlight?
Basically what we discussed, right? Like sometimes this brain area may not even be necessary for this behavior or for where you are with respect to this behavior, right? Like this work by Ben Stovetsky, like if a rat learns this kind of dance of banging on a lever a few times, after a while they don't need motor cortex.
Yeah.
But then there's the more interesting twist that if they learn this along with a similar task, they will always need motor cortex. I don't know if you've seen that paper.
Say that again. If they learn it, no. Yeah, can you unpack it?
Yeah, sorry. So if they are doing like, I think this is a newer version of the task. It's like a piano that rats have like 3 keys and they have to do like, I don't know, A, C, B.
Uh-huh.
And if they learn it with visual cues, and they can either have to figure out the sequence and do it over and over to get rewarded. So they have to figure out this A, C, B, A, C, B. And I think they also train some mice on a visually guided version of the task. And the mice learning the visually guided task always needed cortex. And if they learned both, and this may be a different paper, then they needed cortex also for the one that was not visually guided. So it's not only what you are learning and how you are learning it, but with respect to which other things you are learning. So,
which lab is this out of? I forget now.
Ben Stovetsky.
This is out of the Ovechky lab. Okay. Yeah.
Yeah. It's like their series of papers on rats resting on livers.
I remember it. I have to like look back at it. They publish a lot.
Yeah.
These days.
So maybe I've mixed papers or split papers. I know there's a few that show this, and also like fit with the view that you outlined. That is the way we think about cortex-based ganglia. There is kind of like basically a joint controller. I don't know. It's like it all works together, right? I don't think I am much more sympathetic to that view than the idea that based ganglia selects actions and then cortex executes.
Yeah. Well, it's interesting though. Like reading that current opinions paper, I was thinking about this morning, like because what you guys talk about. And I'm not going to ask you details of it because who knows the details of their own work, their own papers that they wrote years ago. But so what you talk about is like at different time scales, You might need different levels of redundancy with different brain areas. The whole thing is about like our old way of thinking about brain area X is for function Y, and how that's not accurate. And the new way of thinking about it is more this like hierarchy way where there's no privileged top of the hierarchy, but things contribute based on context, etc. And timescales, which is what you guys write about in the paper. But the way that you write about in the paper is timescales in terms of like when you're learning an action or when you're developing from the embryo or evolutionary times, those sorts of timescales. Yeah. But the way that I was thinking about it, and I'd have to go back and read the paper again, I think that it's written in a way where nevertheless, so we were talking about the concept of mechanism. And it's like, I think it's written in a way where like you can use mechanism if you zoom in, let me rephrase this. It's hard to get away from talking about a brain area without talking about its function. In a given context, you're still like, oh, it is a controller, but it's controlling less in this function and it's not controlling in this other context or whatever. But we still ascribe, because it's like we have to ascribe a single concept to a single brain area, and it's like really difficult to not think of it
exactly
like that.
Yeah, exactly. I think we will say like you can map or assign a function to a brain area within these timescales. So maybe it's not the best word, but as you said, it's like you're in development, when you've acquired a skill, when you're acquiring a skill, where you're adapting a skill. And within, basically, we don't talk about that, but the experience of the animal, that is not that obvious, but what you know what to do and how to do and what you will be learning, right? So I think this kind of, the background of this paper is that we were trying to make sense of our own data in the context of all these papers that seem to suggest different things, right? Like it's like, oh, but then now you don't need this brain area to do this. And this is how it came together, right? And of course it's hard because again, we can circle back to our conversation of understanding and having compressed representations. And it's much easier if you think like this area does X, this area does Y, and this area does Z. But maybe that is not how the brain works. So it makes our jobs harder, but in a way it may also make them easier in the sense that when you look at older papers, in the context of your own data or thinking about the brain, you are like, oh, so this is why in this case this happened this way. One concrete example that I think wasn't there is that we have this paper that you sent me on the email from Justice Lab. Is Junchol Park is the first author on mice reaching, grasping, and pulling on a joystick with motor cortex and basal ganglia recording some manipulations, right? And in that paper, we kind of find through different ways, our interpretation of the data is that cortex and basal ganglia jointly control the movement, right? But then Junchol, before I have nothing to do with this paper that I love from Justice Lab as well, so same core first and last author, they showed that if you in the paper that I was describing, let's rewind a second. In our paper, we proposed that it's a joint controller. We were manipulating cortex or striatum before the mouse was given the cue. And we saw that in some trials, the mouse was able to, even if we are inhibiting either cortex or striatum, that could like break free of this inactivation and do the movement, and then movement looked mostly fine. But if you do this when the mice are reaching, if you inactivate cortex, they seem to have a more limited ability to reach away from their body, like from a straight line. And if you inactivate striatum, they seem to undershoot.
Okay.
So the function depends on when you are inactivating kind of the same area.
Yeah.
So it's complicated, but it's like, I think talking about these things is what led us to, let's write something together when there's an opportunity to do it.
Yeah. All right. Okay. So I can't let you go without asking you the question from my lab. Are you ready for it?
I don't think so, but yeah, let's go for it.
You're ready. No, you're definitely ready for it. So, and this is something I should have actually asked you. So this is going back to, we didn't discuss it in depth when we were talking, but I mentioned it in passing, and I don't think we ever unpacked it, and we don't really need to unpack it, or I can describe it here. So this is your work on manifolds, looking at like how manifolds are common between species and between different animals. Like if you, we did talk about it a little bit. If you take like the manifold in one low dimensional space in one animal, you can do like a pretty simple linear transformation and map it on to the manifolds in other animals performing the same behaviors or other species, etc. Okay, so the question is about that, and it's whether you've looked at first, whether you've looked at that in untrained behaviors, but then B, and this is the main question that he wanted to ask is, do you think that you could go into, so you record the neural activity and map out the manifold in one animal. Do you think that you could go into like a naive animal, go in blindly, with the knowledge of the structure of the dynamics and the manifold structure of the representations in the trained animal? Could you go into another animal and like induce via like holographic optogenetic stimulation? Could you know how to induce that same behavior just by knowing the manifold structure? Does that make sense?
Yeah, it's a very fun story. And it feels a bit it feels a bit dystopian. So in that paper, we showed, for clarity, we showed that similarity across individuals from the same species. I should say that Matt Perry, who is the co-senior author on that paper with me, and we've been, working together since he was a PhD student, I was a postdoc. He just posted, I think yesterday on bioRxiv, a paper comparing dynamics and manifolds across mice, monkeys, and humans reaching.
Okay.
So this is a very timely question. The paper, I think it's on bioRxiv as of 1 or 2 days ago. So the story there is quite interesting because what they found, I feel like what's saying we because I didn't do much, but what they found, then I'll stick to that, is that you have the same dynamical rules across all the species, but the geometry is a bit different. And our interpretation of that is that basically the geometry is like what influences the details of the behavior, probably because of, when it's read downstream, right? But just to clarify, but to your colleague's question, I think it's a great question, and I want to believe that would be the case. And I think it would be the case if the animal has the underlying circuitry, right? So
that's the whole point. That's the whole point, right? Is that they do share the same underlying circuitry.
Exactly.
Otherwise they
Okay. Yeah, I guess I meant if one mouse were like the Jimi Hendrix of Miceland, and the other was not very good at using their paws. But I guess in mice this is less of a problem. But I think it would be the case. And actually, we have some indirect evidence of that you can decode across animals based on a decoder, right? So this suggests that it's the same patterns, right? So if you pattern the simulation, you should be able to generate the same population dynamics. And there's evidence of that from Carl Dicerot's group. So I don't think we talked about this paper where they look at. I finished my answer saying that I think it should work, and I'll explain the paper.
Wait, but that's because, but the first thing you said, I just want to make sure I have it right, is that you can like train a decoder in one animal and then you transfer that decoder to a different animal.
Exactly.
And use it. Okay.
And it works. People are doing this, right? Not only us, now there's a lot of work on neural foundation models that is using that, right? That idea. And I think neural foundation models work because of all the preservation in dynamics that we have been discovering, right? Otherwise they wouldn't work. Maybe this is a bit of a tangent, but I think neural foundation models work. I was thinking about this too recently because there's basically similar grammar and vocabulary, if you want, across animals. So probably the dynamic rules and the constraints are preserved across animals, and this is why we
That's interesting.
Yeah. So to go back to this inducing population dynamics, there's this paper from Kardashian's group with Surya Ganguly where they're doing holographic stimulation. Have you seen that paper?
I think so.
It came out during COVID, so I think this is why it's not in most people's radar. And I also only came across it by chance. But what they did is they were doing, fancy stuff. So they were recording from a bunch of neurons in mouse V1, while mice were doing a no-go task. So they were presented with gratings, and to the left would be go lick, and to the right would be no go. And they found the neurons that basically they did PCA and found a manifold that was like going left is go would lead to go and the other to no go, right? Like the two grating
This
directions.
is familiar to me, but I must have read it, but yeah.
Yeah. So you have the two grating directions. And then they found the neurons that were most tuned to each of the gratings. And they stimulated, let's say, the neurons most tuned to go. And then what they found is that the rest of the neurons were doing the same trajectory along the manifold as when the mouse was actually seeing the grating. And the coolest thing is that the mouse went and did what he was or she was supposed to do. That was lick or no lick.
So by stimulating the tuned neurons, the rest of the population followed suit. Is that how to think about that?
Exactly. And the mouse behaved exactly with the same. If you look at the behavior, it's the same psychometric curve, so the same accuracy as when they were seeing the stimulus. So they were basically making the mice, I don't know if we used to say hallucinate the grating, or at least have the, illusion that they had seen the grating, right? And I think it's super cool. And yeah, again, I think maybe they got unlucky with the timing.
That's kind of like, that's like a Hopfieldian kind of thing where if you're, if you're stimulating the right neurons to induce an attractor state or something. I guess that's the simple way to say it.
Exactly, right? And I hadn't thought about it that way, but if you think that this behavior emerges from the interaction of all these brain areas, it means that, just like zapping the right neurons, even if there are a few, in the right way when the animal is kind of in the right context, you can do a lot.
Yeah. So there are these key nodes that you can sort of push, and that transitions the rest of the system maybe into the right full-on dynamical regime. That's
exactly
cool.
Yeah.
So then you wouldn't have to do holographic stimulation, right? You would just you'd have to find the right. But that's weird because it's like going back to the old neuron population doctrine story, right? Where if you stimulate the grandmother neurons, then all of a sudden there's grandma, but the rest of the neurons that we say are important follow suit, and they're also saying it's grandma or something.
I guess. Yeah, I guess the idea is maybe it's not a sparse code, but it's a collective code. I don't know if anyone has said that word, but maybe that is the population code.
Distributed would be the word, right? That people have said.
Exactly.
Collective is another way to say it.
Yeah.
Have you done it in untrained animals? That was the other part of the question.
No, we haven't, because we didn't have any untrained monkeys. And also I think in untrained monkeys, to be honest, it would. Well, no, maybe. Let me think a second.
Sorry, untrained behaviors, not necessarily untrained animals.
I think I'm pretty sure we've tried it out in monkeys reaching. But this is trained, right? Because monkeys reaching for fruit is trained. Yeah. So I'm pretty sure I tried it. We have data from monkeys reaching for pieces of fruit. And I think I tried it during the revisions of one of the papers, and it worked.
Okay. I'll point
but it would be cool to do it in mice, where perhaps things would be different depending. I guess the cool question is whether things will look a bit different because of the time scale the animal is in. So this is how it all comes full circle.
Yeah. All right. Well, I'll let him know. What else? Anything else you wanted to chat about? I appreciate you coming back on with me to do this.
It's fun. Again, we should do this over a beer. It would be much more fun.
For sure.
Or some Vino Verde here. I don't know. I think we've covered lots of things, right? And you actually got me thinking. I was flying back yesterday, and I was thinking about your question about LFPs and epiphenomenal or fundamental and these things.
Oh, cool. That's great. If you're thinking about me in the shower and when you're laying in bed, that's great. That's exactly what I wanted.
Exactly.
Just wait
That is
till
the
like
dream.
we'll be getting beers, and I'll be like, "Oh, hang on, I got to record this," and I'll put like a microphone in front of your face.
Have you done that
No.
before?
No, I've never done that before. I'm not an asshole.
I thought it would be fun because I think with enough beers, most people would agree. So it's not like you're forcing anyone. It's like this is the best idea of our lives. So I guess it
That's true.
depends on the
Yeah,
on
yeah.
that. But
But
yeah,
I mean, the weird thing about like we should have recorded that is that like you said, it's like important to think about these things, and often those sorts of speculative ideas or what people really think will come out like right when I stop recording or something. Someone will say something, they're like, "Oh, like people should have heard that." So that's why this kind of conversation I think is valuable too.
yeah. No, I think and I think like the value of going to conferences is mostly these things, right? Like now we cannot watch talks online, but I think this kind of conversations where you are like relaxed and basically you start like speculating and things, like a lot of ideas come this way, right? And I guess the challenge for all of us is like we are so busy doing is finding the time to think. So I think maybe we should do less doing and more thinking. This is, by the way, I remember this quote from the first time I saw John Krakauer give a talk, and that kind of stuck with me because I think he's right.
Yeah, I mean, there's lots of issues to work out on top of it, but all right. Well, anyway, so I'm gonna stop recording here and hang out for just a second, okay? And Brain Inspired is powered by The Transmitter, an online publication that aims to deliver useful information, insights, and tools to build bridges across neuroscience and advance research. Visit thetransmitter.org to explore the latest neuroscience news and perspectives written by journalists and scientists. If you value Brain Inspired, support it through Patreon to access full-length episodes, join our Discord community, and even influence who I invite to the podcast. Go to braininspired.co to learn more. The music you hear is a little slow, jazzy blues performed by my friend, Kyle Donovan. Thank you for your support. See you next time.